Lisa Bastarache. I come from Vanderbilt University Medical Center, and I've been working for many years now on biobank data and especially extracting research quality phenotypes from the medical record. And this year at CGSI, which I've been to for many years running now, my talk is titled The Real World Phenotypic Presentation of Mendelian Disease. But as is often the case, I regret this talk title as soon as I started putting together my talk, and actually in this case it's sort of misleading, because one of the themes of my talk is that these natural phenomenon we call Mendelian disease, there's actually no one real description of them. They're just descriptions from many different perspectives. So what I'm going to present today is what Mendelian disease looks like from the perspective of the electronic medical record itself. So my talk outline, I have 3 parts to my talk. In general, I'm going to be introducing this thing called the EHR Mendelian Catalog. This is something I've wanted to develop for more than 5 years. And the EMC, as we call it, is a catalog of over 300 different Mendelian genetic conditions and their phenotype profiles, the phenotypes extracted from the medical record in an automated fashion that sort of characterize these disorders. So in my talk, I'm going to explain why I thought the EMC was something worth making. I'm then going to talk about some really aggravating barriers, or really one in particular, to building this resource, with a little bit of a sidebar about clinical genetic testing. And then finally, I'm going to show some examples or applications of the EMC. So first part is about seeing Mendelian diseases from different perspectives. So I think that probably everybody knows what a Mendelian disease is, but just as a reviewer, a Mendelian disease is a genetic disorder that's caused by a single genetic alteration, and that is passed down through families in patterns that Mendel noted when he was studying his pea plants. Oftentimes, when people talk about certain Mendelian diseases, they'll say that they were discovered. Many of these were discovered in the mid and late 20th century. And oftentimes there's even eponymous names for these disorders, like Marfan syndrome, for instance. But really, it's sort of a misnomer to say that these diseases were discovered then, because these disorders have been with human beings since there have been human beings, and they've also been the subject of interest long before we understood ideas about inheritance and when Mendel was doing his work. There's a series of papers that's sort of this crossover between people who are interested in art and people who really are interested in Mendelian disease, who've looked through paintings. These are from the 16th and 17th century, and diagnosed, or posthumously diagnosed, the individuals that are depicted in these paintings. And so here you can see that these, oftentimes these disorders are rendered in such great detail by these artists that these phenotypic patterns that are characteristic of these monogenic diseases are so distinct that hundreds of years later, people can make a pretty good educated guess as to what disorder these individuals are affected by. There's also a lot of indication that people were talking about sort of monogenic conditions in European folklore and fairy tales. There's an often quoted in the CF literature, European folklore or sort of myth, it's actually not a myth, it's true. Woe to the child, which when kissed on the forehead tastes salty, he is bewitched and soon must die. Does anybody know what disease that's referring to, likely? Any Mendelian people here? It's cystic fibrosis. So cystic fibrosis is a disorder, it's autosomal recessive disorder. It's quite common in European ancestry populations, probably more common in non-European than we recognize right now. It's a devastating disease. Probably life expectancy is, early childhood, maybe to late teens, if you have the classical form. And one of the things that it does, because it's a chloride channel, is it makes babies' skin taste salty. And so before we had an ability to actually diagnose these disorders, now actually one of the ways they diagnose them is doing a salt test on your skin to test how much salt is on your skin. But mothers would kiss their babies and realize that they tasted saltier than their other babies, and realize that something bad might happen later. But when we talk about Mendelian genetics as scientists, typically we're referring to the perspective that was really blossomed in the mid-20th century, and that is the perspective of medical genetics. The work of Mendel was forgotten for a little while and then revived, and a number of clinicians who had families that they saw where they had unusual, oftentimes unusual combinations of phenotypes, they used that logic of Mendelian inheritance and started connecting the dots and saying, oh, these individuals are affected by genetic diseases. And so this work, this is a photograph actually of Victor McCusick, who did a lot of work with Amish families that lived near where he was at Johns Hopkins. McCusick is considered the father of medical genetics, and so he spent decades getting to know Amish families and really understanding their family trees, and also and was able to describe a number of different genetic conditions. And this, I'll note, is before, it's after the discovery of what the molecular structure of DNA was, but long before we had any ability to actually do genetic tests. He and many others were working on describing these disorders. The work that McCusick did was recorded in these volumes, initially called the Mendelian Inheritance in Man, now called the Online Mendelian Inheritance in Man, or OMIM, which is a website. It's been a website since the 80s. And MIM, or OMIM, the main purpose of this was started by McCusick, was both nosology, which is a word you may be familiar with if you hang around Mendelian folks enough, which is related to the naming and the delineation of genetic disorders, and nosography, which is the description of these diseases. As I've mentioned before, oftentimes a single genetic defect will lead to a lot of different patterns, phenotypic patterns, and so these volumes contain those patterns as well as the hypothesized mechanism, autosomal dominant, autosomal recessive, or sex-linked. Here we have an example of a phenotypic description that you can call up online on OMIM for Marfan syndrome. This is a disorder that's caused by mutations in FBN1. It's an autosomal dominant disorder. FBN1 is really important for all forms of collagen in the body, and so this is really a defect of collagen, and collagen is important to all sorts of parts of your body. And so you can see here, this is a very abbreviated description from OMIM of what Marfan syndrome is characterized by. There are defects of the eyes, the cardiovascular system, skeletal, and individuals with Marfan's tend to be very tall. Some people think that Abraham Lincoln had Marfan syndrome because he has sort of a characteristic face, long face, that people who are medical geneticists say, you know, he may have suffered from. So, like many people before me, including the artists and the grandmothers telling nursery rhymes and these medical geneticists, I find these patterns to be incredibly compelling. The idea that one single genetic variant can give rise to all of these patterns in this predictable way, such that we could say this person here and this person here have the same thing even before we could do any molecular testing. And I think that this is really important, a huge amount of the knowledge and the foundation for this field, which is, by the way, the foundation for what we do clinically in genetics now. I mean, when people get tested clinically for genetic disease, almost without exception, although polygenic risk scores are starting to get used in the clinic, they are testing for these disorders which were described, started to be described in this project of studying Mendelian disease. So I was really fascinated by these patterns, but I was unable to actually see patients. And this knowledge has been garnered by really careful, close observations of people in the real world studying these natural phenomenon. But my background is in computer science and math, and I don't see patients. I would be a disaster seeing patients. So I wanted to study these from the perspective of data. Fortunately, the Online Mendelian Inheritance in Man, which includes these clinical descriptions, which are really meant to be read and are read by clinicians when they're looking at their patients, they have already been mapped to an ontology called the Human Phenotype Ontology. This is an ontology that was developed for the purpose of studying Mendelian diseases. And so this is from work now. In 2018, we first published this method where we mapped these human phenotype ontologies to something called phe codes, which you all may be familiar with. They're ICD-based codes that take in just the ICD layer of the electronic medical record and give you a couple thousand phenotypes that are sort of clinically meaningful phenotypes that you could derive from these ICD groupings. So what this mapping allowed us to do was to take the clinical descriptions from OMIM and map them to phenotypes that were very easily extractable from the medical record. I feel like I've presented this slide 1,000 times, but we came up with this idea of the phenotype risk score, which is the idea that we could assign a score to each individual in a population. Some who may look a lot like they have Marfan syndrome, some who don't look anything at all like they have Marfan syndrome, they have no features, and others that maybe have some features of Marfan syndrome. And we assign a score to each of these features. It's essentially just weighted so that rare features are upweighted and common features are downweighted. Everybody gets a score, and then we did a lot of positive controls with the phenotype risk score, or PRS, where we just asked, okay, when you look in a large population and you find individuals who are affected by Marfan syndrome, do they have a higher phenotype risk score than unaffected controls? And the answer is yes, reliably so. These disorders really do leave a phenotypic imprint, and this imprint really can be recognized through this process. So the take home here is that we had a method where we could differentiate a group of people who were affected by a condition using only the features of that disease. And that's very important because as we're now looking at these disorders from the perspective of biobanks, and we're starting to do genotype first studies, we're seeing that a lot of these disorders are underdiagnosed. So there are a lot of individuals in the population who have Marfan's or another disorder, another monogenic disorder, and they do not have the diagnostic label. Therefore, if you want to study these individuals from biobank, you can't just say, I'm going to just call people who are affected those who've been diagnosed with Marfan's, because you're going to miss a lot of patients for that reason. So very briefly, the initial application for why we developed the phenotype risk score in the first place was to study rare genetic variants from biobanks. Even since the publication of the first paper, there have been a lot of new databases that have become available where we have whole genome sequencing, whole exome, now whole genome sequencing, and some degree of EHR data linkage, like the UK Biobank, all of us, and a lot of other biobanks around the world. And so the idea is, could we look at these rare genetic variants and determine if they were causative of the underlying disorder? And here you can see, this is a study from eMERGE, which is a group of institutes that sort of share data back and forth. Vanderbilt is one of the institutes. I think some of you guys are probably in the eMERGE network. And we looked at individuals who had known pathogenic FBN1 variants, known benign FBN1 variants, and found that the phenotype risk score is consistently elevated. So this is just one application. But almost as soon as the paper came out, people became very interested in, and then they got me interested in, a second application for the phenotype risk score, which is, I think, a lot more challenging, but maybe more fun too, I guess. Anyways, and that is the idea that could we use the phenotype risk score to find patients who are affected by these conditions and are undiagnosed? As I mentioned earlier, we're now starting to realize that there's a lot of people who actually have these disorders, and some of them have very specific treatments, targeted treatments that are available and very effective. Cystic fibrosis is one of them. And these individuals, especially if they present atypically, can be really difficult to diagnose for clinicians. So the idea is, could we use these scores in a big medical population, find people who have very high scores, and then suggest to their doctor that they get genetically tested and help these patients? And some of the encouraging early results that we saw for this application was that we looked at, in this case, adults who were diagnosed later in life with cystic fibrosis. And we found that many years prior to their diagnosis of CF, they had many of the key canonical features of this syndrome and very elevated phenotype risk scores. So we recently, actually this study took a long time, but we, several years ago, we initiated a pretty small study where we attempted this with the cystic fibrosis phenotype risk score. We took about 1 million patients with medical record data at Vanderbilt and then scored them in terms of their similarity or dissimilarity to the cystic fibrosis kind of clinical description. We took the top 400 scores for the phenotype risk score. 62% of those had already been diagnosed with CF, which is obviously way higher than you would expect. we had extant blood samples on 26 of these individuals and sequenced them, and we did find one, I say potential, but this person very likely has cystic fibrosis. They had 2 pathogenic variants that were heterozygous for. We didn't do phasing, so we don't know if it was compound het, but they're compound het. So 1 in 26 diagnostic rate, which is pretty good. We also found another individual who had alpha-1 antitrypsin deficiency, which is a more common genetic disorder. It's not hugely phenotypically overlapping with cystic fibrosis, but it is characteristic of lung dysfunction, COPD, asthma, bronchiectasis is characteristic of that disorder, so, which is one of the reasons we looked at it. Yeah, please. Yeah, this is BioView. These are de-identified patients. I should mention that too. Is that why you asked? Yeah, we have a biobank at Vanderbilt called BioView, and so we have de-identified medical records linked to blood samples, which is why we were able to do this study. And they are not consented to get results returned, and they're in fact de-identified, so we did not return this result to this individual, unfortunately. But hopefully the work that we do can actually prevent that situation of people not getting diagnosed. But, 1 in 26 is actually pretty good considering the base, just choosing somebody at random is a very, very low probability that you're going to find somebody with cystic fibrosis. But I've long felt that there was a way of improving the phenotype risk score, which is really based on just translating this knowledge from OMIM straight into phenotypes that are compatible with the EHR. And the reason I thought that there was room for improvement is as I got familiar with the way that these disorders were described from the perspective of medical geneticists, I noted that it was really a sort of very specialized, almost niche way that medical geneticists or Mendelian geneticists describe these patients. They use terms that few people in other domains of medicine use to describe their patients in a huge amount of detail. So I mentioned before when I showed you that Marfan syndrome description that it was abbreviated. This is actually, it was really in part for space reason, but this shows just the head and neck features that describe Marfan syndrome in OMIM. And so you have things like dolichocephaly, which is just an elongated head like that, they illustrated there. Narrow palate, which is something that is not necessarily a diagnosis, but just sort of a phenotypic feature. You also have the feature of long face, which is really not a medical condition at all. When you look it up in OMIM, you find the beautiful Aman, who I do not believe has Marfan syndrome. And there's also terms like hypertelorism, that is individuals who have eyes that are further apart than average. And these are not features that are necessarily medical observations whatsoever, and they're only connected to a medical diagnosis once patients are actually found to have a particular disorder. And just to give you a further idea of how incredibly detailed the observations that medical geneticists make, I want to note that in the human phenotype ontology, this ontology that was designed for medical geneticists, there's over 2,400 different distinct terms for abnormal limb bone morphology. So that's your hands, feet, arm and legs. Basically, every bone in your body can either be short or long or too brittle or too hard or abnormally shaped in many, many different ways. And this diagram here is just an example of a number of different abnormalities of the bone structure of the hand. What's really fascinating is that a lot of these structures, they really do, not entirely, but they basically point to specific genetic defects. Like there's some genetic defects that will only shorten like your second, third, and fifth finger pretty reliably. The reason that the medical geneticists came up with all of this incredibly detailed terminology is because that's all they had, 60, 70 years ago when they were studying these diseases, were these clinical observations. They weren't able to do molecular confirmation. That came later. And so in order to really delineate these disorders, they needed to have all of this detail. But when you start looking at individuals who are affected by these disorders from just stuff that's actually collected in the medical record, you realize that this level of detail is not included. And that we need to have, so what we need to do is not merely translate what we can from the OMIM space into the EHR, but we also, I think, have to augment our phenotypic descriptions by using real-world observations in the electronic health record in order to have high-performing, sensitive algorithms to detect these phenotypes. So that really is the idea behind the EMC. what we did is we identified individuals who were affected by these clinical conditions. Again, I'm using the example of Marfan syndrome, and we ran a method called the phenome-wide association study, something I've worked on for my whole career. Hopefully you guys are familiar with it. It's just sort of an inverted GWAS study where we agnostically scan across the medical phenome for associations, in this case with either case control status for having Marfan syndrome. And in the early, when I was looking at this for the first couple of diseases that I was studying, I was really amazed at how clean these PHWASs came out. These really could be like boards questions for medical genetics. Really the canonical features of these syndromes were derivable from this process. Aortic aneurysm, dislocation of the lens, scoliosis, pectus excavatum, these are all really the cardinal signs. But what becomes clear when you compare what you see in this real-world EHR context versus OMIM is that there are differences. So in this chart here, you see that there are 21 features that we both found in OMIM and in the EHR. There were a number of features in OMIM that we didn't see as statistically significant in the EHR, including things like heart failure, emphysema, glaucoma, and then there were several novel associations. Some of these novel associations, I think, were actually just different ways of saying the same thing. For instance, we didn't replicate the feature of glaucoma for Marfan syndrome, but we did find an association with abnormal intraocular pressure. And so likely sometimes there are features that are just really lost in translation from that mapping process from OMIM to the EHR that I think could be rescued. We also found some novel associations, and here's a bit of a cautionary tale because some of these novel associations turn out to be false positives for interesting reasons. So this was really, I think Marfan was the first disease I studied in this way, and one of the reasons I wanted to run this, and I think we are going to succeed in this, is that there's a process called phenotype expansion that people do on these monogenic diseases where they say, okay, we know feature A, B, and C is associated with this disease, but now we think that D is as well as a characteristic feature. So I wanted to do phenotypic expansion, and so when I ran this association study, or the PHWAS on Marfan syndrome, I found that sexually transmitted diseases were significantly associated in Marfans. You know, they were like 20-fold enriched or whatever. And I thought, okay, that's pretty weird, but maybe people with Marfans get STIs a lot. Maybe there's some sort of particular susceptibility to them that's related to connective. It seemed really wild, but of course when you start, if you just look it up in Google, like people will, you can find a paper that you could sort of go from A to B to C. Anyways, but of course I was skeptical of this association and investigated a little bit further. And what it turned out is that Marfan syndrome is actually didn't have a specific ICD code until maybe 10 years ago. And so when patients were diagnosed with this, medical coders would go and try to find a code to say what diagnosis code can I apply to them. And it turns out that Marfan, the eponymous person who this syndrome is named after, also described a syndrome that is a congenital syphilis syndrome. So, back in the day, people always were doing about 15 different things that got their name on stuff. So when people looked up Marfan syndrome to apply the billing code, they found syphilis, and they would inappropriately give a billing code for syphilis to these patients. So I think that this shows, this is just one example, and given the constraints of my talk, I'm not going to be able to get into many more, that I think that taking a purely data-driven approach, you run the risk of actually creating a pretty distorted phenotypic description, and that I think that this process of really creating EHR-enabled descriptions of these disorders has to be some sort of synthesis between knowledge and common sense and what we're seeing in the data. Okay, part 2. I'll make this quick. So why did it take more than 5 years to actually build the EMC, which we haven't published yet? We're really close to submission now on this paper. Well, we ran into a really aggravating problem with when we were trying to make this catalog, because one of the necessary ingredients is that we needed to have a number of individuals that we knew had certain genetic conditions. So we could say these, and I felt it was really important that these individuals had molecular confirmation, that they basically had a genetic test, not just a clinical diagnosis. But the irritating problem is that in the electronic health record, people are getting genetic testing more and more, but there is no clear way to store this information. Typically, when people get genetic testing, it's usually a send-out test. So somebody draws your blood and they send it to Ambry, Invitae, GeneDx, Prevention, et cetera. And what they get back is a PDF report that isn't naturally readable. You can use, so image recognition to do it, and then that gets stuck as a PDF onto somebody's medical record. So there's no sort of machine or readable format of this information. And what that means is that we don't have a big database of who's gotten genetic testing and what those results are, which would be highly convenient for building a series of cases who are affected by this disorder. Yeah.
I work for Hebbring Genetics.
Oh yeah, awesome.
There are ways to collaborate and get a data dump.
Yeah. 100%, yeah. There's a guy, Travis Osterman has been working on that at our institution. And I think and I'm hoping that we're going to be able to do that. And Epic has come up with that genomic module, so there's like a way to store it and things like that. Yeah, I should have mentioned that, and it's a good interjection. One of the issues, though, because what we basically did is we pulled this data for 21 years at Vanderbilt. We looked at 21 years of genetic testing, and Ambry's like a big player in genetic testing. But we found that there were send-out tests to over 100 different unique companies. And so even if we could get it from Invitae, I don't know what's going to happen with Invitae, but maybe you could tell me, Ambry, et cetera, that there's a lot of genetic testing that's getting sent to a lot of different institutions. And all of it's coming back in its own special format and in PDFs. So it's really quite tragic, because I think that, to me, I think clinical genetic testing is a big future resource for researchers who want to study these diseases. And it is an almost entirely untapped resource because of this basic problem with data. Moreover, I know for a fact that this can cause patient harm, because when people have these findings, it can be very difficult for clinicians to actually find them in the medical record or even know a patient has gotten genetic testing. So I think that this genetic testing is enormously important and life-saving, and we need to come up with a solution that, like any other lab result, we have this stored in a structured format. But either way, and I'm already running out of time, so I'll breeze over this. Basically, I initially thought, because I love writing parsers, I was like, I'm just going to parse this, because when people get genetic tests, they get the report back, but then the doctor writes about it. So-and-so got a hereditary cancer panel from Ambry, and they had this finding. So I thought, I'll just parse this out. But it turned out it was just too complicated. It exceeded my ability to write a parser. And so a lot of this was actually manual extraction from the EHR. We're now exploring using large language models to do this type of thing. But basically, this took years of work. It was quite stupid on my part. But then ultimately, we were able to build this database. I'm going to gloss over this. Oh, this is actually kind of interesting, though. So once we did pull all this data together, 21 years of clinical genetic testing at Vanderbilt, we noticed certain trends, like genetic testing is getting more popular. That's not that exciting, but I thought that this was really cool. So this plot here shows the number of unique genetic disorders that have been diagnosed with genetic testing at Vanderbilt per year. And what you can see is that back in 2002, when we started the database, only a couple of different genetic diseases were diagnosed: cystic fibrosis, Huntington's disease, et cetera. At the end of the study period, over 500 different genetic disorders were diagnosed with genetic testing, which I just think is amazing. And this is mostly driven by expanded panels, whole exome and whole genome sequencing, and just our increasing knowledge and ability to attribute, rare patients and their phenotypes to these genetic disorders. Oh, and the other thing is I'm not going to go over this because I really am. I'm going to try to wrap up in the next 5 minutes. Okay, so this shows just the key. We found when we looked at this 21 years that over 100,000 unique tests, over 17,000 molecular diagnoses that we were able to mine out of the medical record. And for the EMC, which is really the major thing I'm presenting in this talk, it's important to note that we had a massive enrichment for rare genetic disorders. So you might be thinking, like, Lise, why did you spend all this time pulling this stuff out of medical records? You just could go to the UK Biobank or all of us and find people with pathogenic variants and study them. But it turns out that these biobanks, particularly UK Biobank, which has like a healthy bias, that population is healthier than average. There are just way fewer individuals, or there are not enough individuals to actually study, and that patients who have these diseases, they tend to get drawn towards academic medical centers like Vanderbilt. And so we did these projections based on Orphanet to ask ourselves, like, how many people would we need in a population-based biobank in order to have the equivalent number of cases for, let's say, Huntington's? And our projection is over 11 million. So we need a biobank of over 11 million individuals to have the same number of patients. So this resource just has huge, like I said, huge enrichment for these rare genetic disorders. And also, so this kind of clinical genetic database has great sample size. I also think that biobanks, there's going to be penetrance issues. So, a lot of these, when we're building the EMC, we wanted to have people who are actually clinically affected by these disorders. And many disorders show variable penetrance, meaning some people are not clinically affected. And also, and this is something that I'm hoping to talk to people about, I'm a little worried about genotyping error for rare variants in these biobanks because, I'll just leave it at that. But, the clinical genetic database, these are like CLIA, take it to the bank, these calls are correct. So for that reason, like I said, I think that reusing clinical genetic data is going to be the future. And in the same way as back in the day when they were doing GWASs, they used to like enroll people in studies and do an MRI or send them to a rheumatologist to do phenotyping. And then we realized, oh, we can get those phenotypes from the electronic health record. I think the same is going to be true for genetic data. I don't think that this kind of thing is ever going to supplant a biobank, which are amazing. But I think it could be really hugely helpful and complementary. Okay, so my final part of my talk, which I'll go over quite quickly, is just talking about some use cases and reasons why I think that that the EMC is going to be very useful in the future. I first want to just go over the steps of what we do to create, use EMC to create these EHR-based disease profiles. First, like I said, we run a PheWAS with case control status of a Mendelian disease as a dependent variable. You have to be really careful about covariates when you're running this model because patients with these disorders get a lot of medical care, meaning they have a lot that is ascertained about them. So you can create a lot of type 1 error there. You run the PheWAS, which is shown here. Then you just do a basic multiple testing correction, and you only pull associations that are positively associated, prune them to make sure they're independent, and then what remains is your EHR-based disease profile. And so, like I said, we've created these for over 300 different genetic disorders. Here are a couple that are shown here. This might be quite small for you guys to see. Here's an example of a different disease that is called TTR amyloidosis. This is a late onset disease. This is a disease that is very difficult to diagnose. It has a huge problem with diagnostic delay and misdiagnosis. We created an EMC-based phenotype, and we already had an OMIM-based phenotype to use for the phenotype risk score. This is a disease that a lot of people actually at Sinai are looking at and are really interested in and have done a anyway. So what we wanted to use the phenotype risk score, back in the day, even more than 5 years ago, to find these patients because we knew diagnostic delay was a really big problem in this disease. But we found that the phenotype risk score just didn't really work that well. If we went to biobanks and we looked at people who had pathogenic TTR variants, they just didn't have elevated phenotype risk scores. And so this was one of the diseases that I was really hoping, like, if we could use a profile that is sort of a blend between OMIM and this EHR stuff, do we actually get better performance? Are we able to detect these patients from biobank? And this is just from last week, some prelim data that we generated. The answer looks like yes. It looks like using the EMC version of the phenotype risk score enables us to actually discriminate between individuals who carry the TTR pathogenic variant versus not. And I guess I made my talk too long, sorry guys, but in the waning moments, all of the stuff that I've shown right now actually just uses very high level coded data, ICD data from the EHR. But as we were putting together the EMC, I started to get really excited about the idea of using different modalities of data that's collected in the EHR to start profiling these diseases. Like, why just stop at ICD codes? And so some of the preliminary work that we've been doing there, which I think is really encouraging, is using things like, for instance, all the height and weight measurements that we all have to do when we go to the doctor. Can we repurpose them and find differences in the way that individuals with certain diseases, in this case, again, Marfan syndrome, how they grow as children? And the preliminary results here are really encouraging. Here we show growth curves across a whole population, which are pretty closely conformed to what WHO or the CDC publishes as like the normal growth curve for children. And then in orange or yellow, you can see the Marfan syndrome growth curves, which are consistently higher than average. This is a well-known and established feature of Marfan's. And we're not the first people to think about doing disease-specific growth curves. There are growth curves that clinicians use for, let's say, patients with trisomy 21, because these growth curves, anybody who has kids, you realize like this is a big important way of tracking kids' health, is to say, are they growing normally? But if a kid has trisomy 21 or Prader-Willi or Marfan's, then their normal is going to be different than the population normal. But given what we've seen with this growth data, I think it's really promising to be able to create a lot more disease-specific growth curves for clinicians to use, and also to maybe gain new insights into differences in the way that individuals with these diseases grow as children. And very finally, I work a lot with Dan Roden, legend, love the guy. He's a cardiologist, and he does a lot with long QT syndrome and other arrhythmias. And so I was interested in, could we take all the ECGs that are done? We have millions of them in patients, and look for differences in these ECG measurements. And that was probably pretty hard to read, so I zoomed in here. And again, this is really prelim data that we just generated in the last couple weeks. And what we see is that if we take the QTC measure of individuals affected by genetic disease and compare them to the population, we see that we do reproduce like these known long QT syndrome, and actually TTR amyloidosis is known to prolong the QTC interval as well, and we're able to replicate those associations. And then we had this association down here from multiple endocrine neoplasia, which is associated with a shorter QTC interval, and I was at first like, oh no, this is obviously a false positive, this isn't working very well. But then I looked it up in HPO. There are a couple of diseases, I think Brugada syndrome and short QT, that are associated with a shorter QT, but also multiple endocrine neoplasia type 1 is also known to have a shortened QTC. So this is actually a replication, which I think is really exciting. And again, these measurements of ECG are often taken serially, so I think that this opens up a whole new window of sort of looking at these genetic disorders, not just from the perspective of high-level EHR billing codes, but also from the perspective of growth, from the perspective of ECGs, et cetera. And I'm hoping that first at Vanderbilt, and then hopefully, across many different institutions, we can start creating these catalogs that can create these descriptions that can be used either computationally or by clinicians to sort of enhance our understanding of these diseases. When I presented this data a month ago at the NIH, I think it was Mark Williams raised his hand and said, I work at the Undiagnosed Disease Network. He's like, we need a diagnosed disease network, and I completely agree. We need to have institutions that are academic medical center where most of these patients receive care. We need to create a network that can share EHR data so we can create these profiles, because I really think that it will be a genuine advance in our ability to diagnose these patients and give them better prognostic information once they do get diagnosed. So with that, I'll thank my colleagues and my lab, and I'll take any questions. I think I went a bit long, so I'm sorry.